we are going to talk about pancreatic secretion. Now, the point you need to understand about pancreatic secretion is this, that you need to know something about pancreas first, okay? Because we will be talking about many things, okay? We will be talking about many things, but you should always understand that what is pancreas first. Now listen, pancreas is actually an endoexocrine gland. That means it releases hormones as well as it releases enzymes, right? Now, this is the first very important point you need to understand, that it's an endoexocrine gland. That means it's a mixed gland. Clear? It's a mixed gland. This is the first thing we must understand. But another thing we must understand is this, that whenever we are talking about pancreatic secretion, the first thing we need to understand is this, that we are going to talk about the anatomy of pancreas. We are going to talk about the anatomy of pancreas, okay? We are going to talk about the anatomy of pancreas. Now, whenever we are going to talk about the anatomy of pancreas, we must understand that pancreas, okay, we will have a detailed class of anatomy, don't worry. So, suppose this is your pancreas. So, this is the main, ductal system of pancreas, okay. This is the main ductal system of pancreas, okay. Suppose this is your pancreas body, okay. This is the body, this is the tail, okay, and this is the head of the pancreas, and this is the main duct of pancreas. Now, this duct of pancreas is actually known as duct of Wilson. Duct of Wilson, okay, it's known as duct of Wilson. Now, it's a duct of this duct is actually the primary duct. This is actually the primary duct of pancreas. This is actually the primary duct of pancreas. Primary duct of pancreas, okay. This is the first thing we need to understand in, anatomy section. Clear? Okay. Okay. We are capital guys. Okay. Now listen. So, okay. So this is the primary duct of pancreas. Now there is a secondary duct of pancreas as well. There is also a secondary duct of pancreas. Okay. The secondary duct of pancreas is known as duct of Santorini. Duct of Santorini. Okay. Duct of Santorini, which is a secondary duct. Okay. Which is a we are again what? Capital guys, right? Secondary duct. Okay. Clear? So this is duct of Santorini and this is duct of Wilson, which is the primary duct of what? Primary duct of pancreas. Now, a very interesting point we again need to understand here in this particular thing is this, that this is actually the main flowing duct, okay. From this duct actually every secretion is coming out, okay. Secretion means here in this case enzymes, okay. In this case enzymes and HCO3 minus, okay. HCO3 minus and enzymes, okay. Clear? Enzymes, HCO3 minus, all these things comes out of the duct of Wilson, okay. Now we need to talk about some inner anatomy of our pancreas, okay. Now listen, pancreas is an acinar gland. Pancreas, if we talk about pancreas, then pancreas is an acinar gland. Is an acinar gland. Okay, is an acinar gland. Clear? Pancreas is an acinar gland. Now, what is the meaning of the term acinar? We will talk about that today, right? But before that, we must understand the inner anatomy of pancreas first. Okay, inner anatomy of pancreas first and foremost. Clear? Now, when we are talking about the anatomy, now the internal anatomy of pancreas is, listen, so suppose this is your main duct, okay, suppose this is your main duct, okay, clear, and suppose this is your interlobar duct, this is known as interlobar duct, okay, and this interlobar duct actually, this interlobar duct actually divides, and this is called interlobar ducts, this is called interlobar ducts, okay, this is called interlobar ducts, and these, the branched interlobar ducts are known as intralobar ducts, okay. I am sorry, this will be interlobular ducts, sorry, interlobular ducts, sorry, okay. Now listen, each and every intralobular ducts enters a particular lobule which looks like this cherry, okay. Now I will be drawing the details of this particular, lobule. So this is called lobule, okay, this is known as lobule, clear. Now what happens is this, that this lobule, now we will have to make a transverse section of this lobule, and then we will see some very basic things, okay. Now listen, which is very important as well. Okay, now listen, the division of these intralobular ducts, these ducts are called intercalated ducts, okay. These are called intercalated duct. Now this intercalated duct actually enters an achinus, clear. This intercalated duct actually enters achinus, okay, clear. Now we will be drawing a bigger picture of acini, okay. Now listen, I have already said that pancreas is an acinar gland, okay, that means its main function lies with acini, okay. Now so this is suppose your acini. In simple term, if you draw, suppose this is a duct, okay, acini looks like this, okay, it is surrounded by many cells, okay, it is surrounded by many cells, okay. Now pancreas is an acinar gland, okay. Pancreas is an acinar gland as well as salivary. Salivary is also an acinar gland. So what's the difference? Both are acinar glands. So can you tell me what's the difference? The difference is that both pancreas and salivary, though they are acinar glands, but there is a specific difference, and the difference is the duct in pancreas enters the acini, but the duct in salivary glands never enters the acini, and this is histologically very important, okay. They never, so this is actually salivary gland and this is actually pancreatic. Okay, so this is salivary acini, this is pancreatic acini. That's the very basic difference you need to understand. Okay, now listen. These are actually these are actually the assigner cells. These are actually the assigner cells. Okay, these are actually the assigner cells. Okay, and now listen with all your attention possible on this earth. These cells which line the duct, that is intercalated duct, these cells which line the intercalated duct, these cells are called ductal cells. These are called ductal cells. Clear? So these cells which line the duct, they are called ductal cells. Okay. Now listen with all your attention. Ductal cells secrete HCO3 minus. Okay. And assigner cells are actually the factory of enzymes. Okay. They are the factory of enzymes. Clear? That means all the enzymes which are secreted by pancreas are actually secreted by assigner cells when proper stimulus comes. Clear? When proper stimulus comes, assigner cells actually secrete what? Enzymes, and ductal cells, when they get proper stimulation and proper signal, they secrete HCO3 minus, which is very much essential for neutralizing the acidic food content, that is chyme, which is coming from what? Stomach. Clear? Am I clear to everyone? So this is the basic anatomy you need to understand before we get on with our journey with respect to pancreatic secretion. Before we go into the details of pancreatic secretion and all these things, now listen. What is the composition of pancreas? What is the composition of pancreatic secretion? What is the composition of pancreatic secretion? Anyone? Now can you tell me that what is the daily output of pancreas with respect to enzymes and all these things? What is the daily output of pancreatic secretion? Listen, the daily output of pancreatic secretion is 1.5 liters approx. Clear? 1.5 liters of enzymes and all other things are mixed and secreted every day by your pancreas. Clear? Now what is the composition of pancreatic juice or pancreatic secretions? What are their compositions? Anyone? What are the compositions of pancreatic juice or whatever it is? Composition? What is the composition of pancreatic juice? Listen, they are made up of amylases. Amylases are enzymes which break down the carbohydrates. Okay, they are made up of proteases. So amylases are carbohydrate digestive enzymes, proteases are protein digestive enzymes, and they have lipase. Okay, now listen. So these are the three categories of enzymes. Okay, composition of enzymes. If we talk about anything else, then we will be mentioning this here. Okay, so composition of enzymes. So what are the types of enzymes present in pancreatic juice? So the compositions are amylase, protease, lipase. Clear? Now can you tell me what is the most abundant enzyme? Which class contains more enzymes? Which class contains abundant enzymes? Listen, proteases are abundant in your pancreatic juice. Clear? Proteases are abundant. Okay, in your pancreatic juice. Therefore, you need to protect your pancreas and you need to protect your GI tract from autolysis. Clear? Now, a very basic mechanism how our pancreas does this job is they secrete enzymes which are not active. our GI secrete enzymes which are not active. Clear? The secret enzymes which are not active, that means they are secreted as what? Proenzymes. Okay, they are secreted as proenzymes. Clear? Apart from enzymes, other composition are ions, okay, and many other enzyme inhibitors and others. Means very simple, there is trypsin inhibitor which is secreted by pancreas, trypsin inhibitor, okay. There is glycoprotein 2, or GP2, okay. Now in this heading, that is ions, got Na plus, Cl minus, H2O, HCO3 in abundance, okay. Pancreatic amylase, or to be very specific, pancreatic alpha amylase, okay. Pancreatic alpha amylase. Among proteases there is trypsin, obviously they are secreted as inactive enzymes, trypsin, chymotrypsin, procarboxy. So other enzymes include, lipases, that is pancreatic lipases and colipases. Colipase is actually not lipase, it helps in the action of lipase, pancreatic lipase. There are RNases, okay, which digest RNA, and there are DNases which digest DNA. Okay, clear? There is procarboxy peptidase A and B. Okay, you all know these things. We will be talking about details in details. We will be talking about in details with respect to procarboxy peptidase A and B, chymotrypsin, trypsin and their action, and how they cut actually a protein in biochemistry class. Clear? Okay. Now we will be discussing the mechanism of regulation of pancreatic secretion. Clear? Now, as you all know that in every secretion there are 3 stages, right? Okay. Stages of pancreatic secretion. So there are many stages of pancreatic secretion, right? Wrong. There are only 3 stages of pancreatic secretion, right? That is cephalic phase, there is gastric phase, okay, and there is intestinal phase. Okay. Now, before we actually move into the details, there will be a quick review. There will be a quick review on all these phases. Okay, there will be a quick review for your USMLE. Okay, now listen. Now, can you answer this simple question? In which phase secretin is secreted? Intestinal phase. Okay. Gastric phase. Can anyone say in which phase secretin is secreted? Somebody was saying in the cephalic phase. He or she lacks a brain. Okay, now listen. Secretin is secreted in the Intestinal phase. In which phase CCK-PZ, cholecystokinin pancreazymin is secreted. Now you get this. Very good. CCK-PZ is also secreted in the intestinal phase. So what I am trying to hammer into your mind is this, that listen, everybody can take this in the brain, right? If you take this into mind, then you can actually go for research also, okay, after you have done your MBBS. Now listen, the first thing we need to understand is this, that what I am trying to deliberately hammer into your mind is this, that both secretin and CCK-PZ, which are mainly associated with these two, words, are actually associated with what? Obviously pancreatic secretion. Now, all these hormones are of immense importance. Now, can you tell a fun fact about secretin? Fun fact about secretin is this, that they are the first discovered hormone. This is the fun fact. Okay, they are the first discovered hormone. Clear? Now listen with all your attention. A very interesting part you need to understand here is this, that in the gastric phase you get another hormone, that is gastrin, right? In the intestinal phase there is no hormones involved. There is no hormones involved. Clear? There is no hormones there is no hormone which is involved in the cephalic phase. Clear? But in the gastric phase there is gastrin. In the intestinal phase there are two hormones, secretin and CCK-PZ. Again, fun fact. Secretin is secreted by which cell? CCK-PZ is secreted by, listen, secretin starts with S, it is secreted by S cell, and CCK-PZ is secreted by I cells. Clear? That's the very basic thing you need to understand. So, what are the takeaways in this small review? That is, we will be talking about in details, don't worry. That is, the most important secretory events which are occurring during the pancreatic secretion occurs during intestinal phase. Clear? Now, in the intestinal phase, two hormones actually regulate the intestinal phase, that is secretin and CCK-PZ, right? Now, secretin is secreted by S cells and CCK-PZ is secreted by I cells. Clear? Now, another fun fact. CCK-PZ, where does this CCK-PZ work? Where does this CCK-PZ work? First, listen. S cells, okay, let me create some space here. Okay, let me create some space here so that you people can understand. Listen. S cells give out what? Secretin. Right? S cells give out secretin. Listen. Secretin actually acts on pancreatic ductal cells. That means indirectly secretin is associated with secretion of HCO3 minus, not the enzymes. Clear? So secretin is associated with the secretion of HCO3 minus. Clear? Okay. Now, let's talk about I cells, obviously. Clear? Here is your I cell. Okay. I cells secrete CCK-PZ, cholecystokinin pancreazymin. CCK-PZ now acts on acinar cells, right? Acinar cells. Pancreatic acinar cells. And these pancreatic acinar cells actually secrete enzymes. Am I clear? So this is the basic takeaway message you must understand before we start this whole discussion. Clear? Before we start this whole discussion, this is the basic takeaway message you must understand first. Clear? Now, let's start in details one by one what is happening everywhere. Okay, in the intestinal phase, in the gastric phase, and in the in the cephalic phase, in the gastric phase, and in the intestinal phase. Clear? So in the cephalic phase, what happens is this. In the cephalic phase, what happens is this, that smell, okay, let me write it here, smell and sight of food, not taste, sight of food actually activates all these things. Smell and sight of food activates particular region in your brain centers, specifically dorsomedial hypothalamus, and this was at first proven in dogs. Okay, dorsomedial hypothalamus plus limbic area and all these things. Okay, now when dorsomedial hypothalamus and limbic area are active, when they are now active, they actually send efferents. They send efferent projections. They send efferent projections by the help of vagus nerve. Okay, clear? And what happens is this, that this vagus nerve secrete acetylcholine. Okay, now this acetylcholine actually binds with a receptor. Okay, acetylcholine now binds with a receptor which is present here, and this receptor is called M3 receptor. This is called M3 receptor. Clear? Okay. Now listen, when acetylcholine binds with M3 receptor, clear? Now this M3 receptor is coupled to GQ. Okay, GQ, and after that, you know what happens, that due to the activation of GQ, okay, you know, PIP2 is degraded by phospholipase C beta, and IP3 and DAG is produced. Where? In this cell. Okay, as the space of the cell. Okay, let me do one thing. Let me, let me make these cell a bit bigger. That means a magnified view of this cell will be helpful, I guess. Suppose here is your, acetylcholine, and it is coupled to GQ. Clear? Now this GQ activates phospholipase C. Now this phospholipase C acts on PIP2, which is present in the cell membrane. Okay, it's a cell membrane. Phosphatidylinositol diphosphate is a phospholipid. Okay, now this is converted into IP3 and DAG, right? Now IP3 acts on specific channel proteins. IP3 acts on specific channel proteins which secrete calcium, and this calcium is helpful for releasing enzyme rich vesicles. Okay, so enzymes, proenzymes actually, proenzymes will now come out. Clear? Okay, and DAG will also phosphorylate these specialized proteins like SNAP25 and all these things, and therefore this vesicle will move towards the cell membrane, and therefore these proenzymes will be released. Okay, and it will come out by the help of this duct. Okay, clear? So this is actually cephalic phase. Okay, so the main stimulus for cephalic phase is smell, sight of food and all these things. So, and what are the pathways involved in cephalic phase? Dorsomedial hypothalamus, and what are the centers activated during cephalic phase? Dorsomedial hypothalamus, limbic system. Okay, and which nerve actually activates this receptor? That is vagus nerve. And what is the name of the receptor? M3 receptor. So there are 4 things you must understand before we move on to next phase. That is, number 1, what is the stimulus? What is the stimulus? Number 2, what are the neuronal centers activated? That is dorsomedial hypothalamus and limbic centers. Okay, and number 3, which nerve actually is activated when cephalic phase is active? Clear? Vagus nerve and the receptor name. That's all. Clear? Now listen. Cephalic phase is an entirely a neural mechanism. It's an entirely neural mechanism. It has nothing to do with other hormonal secretion or something like that. Now listen. Cephalic phase ends when all these stimuluses are gone. That means when smell of food and sight of food are gone, then this cephalic phase will stop. Clear? Now listen with all your attention. So how to stop? If I write how to stop cephalic phase, for example, what will be your answer? Just withdraw the stimulus, right? Just withdraw the stimulus. Now there is another way. Doctors have actually understood how you can actually diminish the effect of cephalic phase without even diminishing the effect of stimulus. What is the chemical name? Atropine. Atropine is an anticholinergic drug. Okay, atropine actually inhibits acetylcholine action. Okay, and binding of acetylcholine with M3 receptor is actually blocked by atropine, which is a classical anticholinergic drug. Clear? It is blocked by atropine. Clear? So what are the 4 things you must remember? First, what is the stimulus? Number 2, what are the neural centers which are activated during cephalic phase? Number 3, what is the name of the nerve which is actually taking all the information towards the pancreatic assignee? Vagus nerve. Now number 4, what is the type of receptor? M3 receptor, and it is coupled to GQ. Clear? These are the 4 things. Now another question, how to stop cephalic phase? Chemically, atropine, by the use of atropine. Clear? By the use of atropine, you can actually what? Chemically inhibit cephalic phase. Okay. Now in the gastric phase, what happens is this, that whenever food particles enter the antral area of your stomach, this is the antral area. Okay, antrum of your stomach. Okay, when food particles enter the antrum area, then from here, then in this antrum area, there are certain cells called G cells, and they start secreting gastrin, which is a hormone. Okay, gastrin, which is a hormone. Clear? Into the bloodstream. So your blood is having now gastrin. Now this gastrin actually, by the help of your blood stream, travels and acts on receptors, okay, present on the, oh sorry, no, gastrin now travels by the help of blood and acts on receptors which are present on the acinar cells. Now, when gastrin comes here, there is a receptor which is already, waiting for gastrin, and this receptor is called CCK2 receptor. Clear? This receptor is called CCK2 receptor, and this is actually gastrin receptor. Okay. Now, there is a very much similarity between the acetylcholine receptor and gastrin receptor. Why? Because CCK2 and acetylcholine receptor both are GPCR. Clear? Both are GPCR. That's it. So the first point we need to understand is this, that gastrin acts on CCK2 receptor when it is secreted by G cells of stomach, and where G cells are present in the antrum of stomach. Clear? Okay. Now listen. Gastrin acts on CCK2 receptor, and CCK2 receptors are coupled to GQ. I think now everything you know what will happen, because I have previously discussed in the cephalic phase what will happen when this ligand binds with this receptor which is coupled to GQ. PIP2 pathway or GQ pathway will follow. Clear? Phosphatidylinositol pathway will follow, and there will be subsequent release of enzymes. Now, if you don't understand this particular part, okay, go to the cephalic phase, then you will understand what is happening in this particular cell. Clear? The molecular mechanism. Okay. Suppose there is food in your stomach. Okay. Now this food is having what? Peptones and peptides, small peptides, right? And this will now initiate a reflex. Okay. And your brain actually get information regarding these peptides and peptones and all these things. Now your brain now comes back to this. Okay. Now your brain will fire a neuron, and from the terminal end of this neuron a peptide is secreted called GRP, or GRP stands for gastrin releasing peptide, GRP. Clear? Now this GRP acts on these cells. G cells, and then these G cells also secrete gastrin. So there are two mechanisms by which gastrin is secreted. Number one, when food enters antrum portion of your stomach, then gastrin is secreted by the help of G cells. Another thing, this is the first mechanism, and second mechanism is this, that whenever there is food in your stomach, then there will be an efferent pathway which leads to the brain, and this activates the brain centers which are associated with digestion and absorption. Now brain will again fire another neuron, that is this is the efferent pathway, and this efferent neuron will now secrete GRP, or gastrin releasing peptide, and gastrin releasing peptide receptors are present on the G cells, and G cells will now secrete gastrin, and gastrin will do its job. Clear? Okay. Now this particular portion, this particular thing is a reflex action, right? This particular pathway is a reflex action, and this reflex is called vagovagal reflex, because this is also a vagus nerve which is going into the brain, and the efferent nerve which is coming down is also a vagus nerve. That's why it is vagovagal reflex. Vagovagal reflex. Am I clear? Clear? So this is vagovagal reflex. Okay. All right. Now we are going to look at the intestinal phase of pancreatic secretion. Now listen. So at first we will be talking about two hormones, that is secretin and cyclic PZ, right? Now listen, whenever acidic food enters your duodenum, what happens is this, that acidic food actually contains what? H plus. Acidic food actually contains H plus. Now listen with all your attention. When this acidic food actually enters this area, this is actually sensed by a cell called S cell. You know what is S cell? S cells are secretin cells, right? So S cell now secretes secretin. Okay, S cell now secretes secretin due to the acidic content of our food, that is chyme, right? Now what happens is this, that this secretin actually enters the bloodstream. As it is a hormone, it enters the bloodstream and its receptors are present on the ductal cells. Clear? And its receptors are present on the ductal cells. Now this secretin acts on receptors which are present on the duodenal cells. Clear? Which are present on the duodenal cells. Now a very interesting point you need to understand here is this, that when this secretin interacts with this receptor, now what happens is this, cyclic AMP concentration increases inside this cell. Okay, cyclic AMP concentration increases inside this cell. Now what is the ultimate function of these ductal cells? What is the ultimate function of these ductal cells? Ductal cells will secrete HCO3 minus, right? Ductal cells will secrete HCO3 minus, right? Now a very interesting point you need to understand here is this. Now listen, a very interesting point you need to understand here is this, that HCO3 actually is coming to neutralize this acid, right? HCO3 is coming to neutralize this acid. That means it's a feed forward mechanism. Okay, clear? Now listen with all your attention. So now you know why this HCO3 minus is coming. Okay, now what happens inside this cell and how this HCO3 is produced inside these ductal cells? What are these? These are nothing but your ductal cells. Okay, these are nothing but your ductal cells, right? Now what happens inside this ductal cell is very, very interesting. Okay, what happens inside this ductal cell is very, very interesting. Now listen, suppose this is your ductal cell. Okay, suppose this is your ductal cell. Okay. Suppose this is your ductal cell. Clear? Now suppose this is the apical part of your cell, and this is the basolateral part. Okay, basolateral part. Okay, now what is apical part in this case? Now listen, this is the apex, apical. Okay, this is the apical part. Clear? And this is the basolateral part. So in this diagram I am referring this like this, and this is the apical portion. Now listen with all your attention. A very interesting part you need to understand here is this, that apical part, okay, apical part, you must remember this very basic thing. Apical part and basolateral part actually have very different kinds of proteins, so that HCO3 minus can be produced. Now listen, in the basolateral part, CO2, let me sacrifice this particular thing so that you can understand. Okay, CO2 enters this cell together with H2O, and there they form H2CO3. Okay, and this is actually occurring in presence of an enzyme called carbonic anhydrase, the fastest acting enzyme in the world. Okay, now this H2CO3 breaks down into HCO3 minus and H plus. Now this H plus goes out of the cell by the help of a protein. Okay, this H plus goes out of the cell by the help of a protein. Clear? Okay. Now this is very interesting. This is actually an exchanger protein, and therefore it will exchange H plus with another ion, that is Na plus. Clear? H plus with Na plus. Clear? Now another thing will happen, that is this Na plus will be thrown out by the help of this, and potassium will come in, and now you know what is the name of this protein. It is actually utilizing, ATP. Okay, it is actually utilizing ATP. You know what is the name of this protein? What is the name of this protein? This is sodium potassium ATPase. It is, sorry, oh my god, sodium potassium ATPases. Okay, clear? And this is sodium hydrogen exchanger. Okay, this blue one is a sodium hydrogen exchanger. Okay, now potassium moves in and sodium moves out. It's so simple. Okay, now in the apical surface, what happens is this: HCO3 minus escapes with the help of this protein, and chloride also escapes by the help of this protein. So this is actually HCO3 Cl minus transporter. This is actually HCO3, that is bicarbonate chloride transporter. Bicarbonate chloride transporter. Clear? Now a very interesting part you need to understand here is this, that this particular protein has a very much important significance in our CFTR. Sorry, this particular protein is very much significant with respect to a disease called cystic fibrosis, because this particular protein channel is called CFTR. What is the full form of CFTR? Anyone? What is the full form of CFTR? Cystic fibrosis transmembrane conductance regulator. This is the full form of cystic fibrosis CFTR channel. Okay. Now, what is the importance of this particular channel? That is, it takes out both chloride and bicarbonate. Right. Now, a very interesting part you need to understand is this: it takes out 2 bicarbonate and 1 chloride. Okay. That means this cycle will have to repeat twice in order to take 2 bicarbonates. Okay. Now, you know what is the problem in cystic fibrosis disease when it comes to this particular channel? The problem is this, that there is a specific type of deletion. Okay, that is delta F508. You know what is this? Delta means deletion. We will talk about this later, don't worry. Deletion. F for phenylalanine. This is a single letter code. And 508 is the position of amino acid, phenylalanine. So when this mutation occurs, there is a deletion of phenylalanine in the 508th position of this protein, and therefore this is important for you to understand, and therefore this protein is non-functional, and therefore bicarbs are present in the cell obviously, but they cannot come out of this cell. Clear? And therefore in cystic fibrosis patients you will see 2 things. First, they have a lung disease. Second, they have a severe pancreatitis. Clear? They have a pancreatic inflammation. Clear? These are the 2 things you need to understand. Now let us come back here, that what this secretin was doing, right? What this secretin was doing. Actually secretin receptors are present here in the basolateral membrane, and when secretin binds with this receptor, cyclic AMP concentration increases, and therefore protein kinase A is activated. protein kinase A is activated, and therefore when protein kinase A is activated, phosphorylation of CFTR takes place. Phosphorylation of CFTR takes place, okay? And when phosphorylation of CFTR takes place, what happens is this, bicarbs are coming out. So this is the importance of secretin, that is, it helps in the passage of bicarbs, not in the formation of bicarbs. Clear? In the ductal cell of pancreas. Clear? So this is the first mechanism by which your actually pancreas prepares for the digestive phase, and by neutralizing H plus, okay, by neutralizing H plus. Now what is happening in this particular portion, that is to the apical cells, okay, what is happening in the apical cells, we must understand this now. Now we will be talking about the role of acinar cells, okay. Now when lipids enter the duodenum, then I cells are activated. I cells are activated. Now, a very interesting part you need to understand is this: when I am talking about lipid, this doesn't mean triglyceride. Actually, triglyceride, listen with all your attention. Triglyceride actually never activate I cells. Clear? They do not activate I cells. That's the very basic thing you need to understand. But when triglycerides are converted into fatty acids plus glycerol, actually not glycerol, monoacyl fatty acids more than 18 carbons can actually activate the secretion of CCK, okay, clear. Now when CCK comes into blood, it then reaches the apical cell, okay, it then reaches the apical cells, okay, it then reaches the apical cell, okay, it then reaches the apical cell. Now a very interesting part you need to understand here is this, that when it reaches the apical cells, then the basolateral membrane of apical cells actually have receptors for this CCK, okay, and the name of this receptor is actually CCK2 receptor, okay, CCK2 receptor, okay, CCK2 receptor. Now very interesting part you need to understand here is this: CCK2 is coupled to GQ. Now when GQ is active, then it will split, it will activate PLC beta, right? PLC will act on PIP2 to give IP3 and DAG. DAG will activate, now listen, DAG will activate protein kinase C. Protein kinase C will now activate or phosphorylate, actually will phosphorylate those proteins which are associated with docking, priming and fusion of these vesicles containing enzymes. So these vesicles which are present in the apical cells contain digestive enzyme, okay, they contain digestive enzymes, clear, they contain digestive enzymes, and when these digestive enzyme containing vesicles are released, okay, these enzymes, these proenzymes actually, these proenzymes now comes here, and by the help of enzymes present on the brush border and all these things, these proenzymes will be activated into proper enzymes, right? Now listen, everybody knows these steps and everybody knows these mechanisms, but very few people know what I am going to say right now, okay? Now listen, CCK, now listen, CCK also acts on neurons, okay? CCK also acts on neurons, okay? Now listen, what happens is this, that CCK, okay, acts on neurons which are actually bipolar. So CCK acts on neurons CCK by the help of blood acts on neurons which are bipolar in nature. Which are bipolar in nature. Now, when CCK acts on these receptor, okay, then brain gets activated, right? And via vagus nerve, now listen with all your attention, via vagus nerve, brain will activate via vagus nerve, brain will now activate both apical as well as ductal cells. Clear? This is very interesting. Okay, now apical cells have receptors for what? Apical cells have receptors for acetylcholine, ACH. Clear? And these receptors are called M3 receptors. M3 receptors. Clear? And these are also coupled to M3 receptors. Okay, M3 receptors. So this is how CCK-PZ acts as a hormone so that proenzyme activity can be increased. But on the other hand, it acts as a neuromodulator. So that both secretion of HCO3 minus as well as proenzyme is increased, okay, from pancreas. Clear?